Hello, everyone. Thanks for joining this invite talk. So, on the behalf of CSCR, it's my pleasure to invite and introduce Dr. Seetharaman. So, he's Associate Professor in Department of Pharmacology, UTHSC, and also a Director for Structural Biology at UTHSC. So, to describe short about him, so he did his PhD in University of Madras Guindy campus in the field of biophysics. Then in 1998, he moved to Canada, Toronto, and did his postdoc for 6 years. So basically, his work is mainly focusing on characterizing the proteins using X-ray crystallography. Then after that, he moved to Brookhaven National Laboratory in New York, and he worked for almost 15 years. And then during those 15 years, he characterized more than 500 proteins and put it in protein database. So then he moved to St. Jude Children's Research Hospital for 5 years as a senior scientist. Then recently, he's now in UTHSC. So most of his work is like validating the proteins and characterizing the proteins. So he has got a chance to work with Nobel Prize winner last year, David Baker. So he resolved some of the proteins for him. That is kind of integral part for our achievements for winning the Nobel Prize. So with this, I will invite Dr. Seetharaman, and I'll thank for him for joining this session. So please come on.
Thank you, Darash. Good morning, everyone. Happy to be here and actually proud to be part of this seminar series. Last week, I came from US and we got together for about 5 years at St. Jude. And I called him, I said I wanted to come and visit him. And he also asked me to come and give a talk. Basically, I'm a structural biologist, but structural biology is very basic fundamental research. I'm going to talk a little bit about that, how it kind of validates everyone else's research. So what I do at UTHSC is, I forgot to expand that, University of Tennessee Health Science Center. So that's the medical school, large medical school at the University of Tennessee that has mainly MD program, which is like MBBS here. MD program, and we have dental program, School of Nursing, School of Pharmacy, School of Pharmacology. It's a huge school with different colleges and departments. So I established a course center there that's called Structural Biology Course Center. I'm going to explain what is that a little bit. I'm not going to talk too much about that. Structural biology is a subject where you need to have some knowledge in physics, mathematics, and programming, because it has two parts. One is the basic biology part, where you work with proteins, purify, clone, get your protein ready. Once the protein is ready, you have to do the structural work. That's more computational experimental part. Okay, so let's start. I'll talk a little bit. So we can say that for human, there can be two states. One is healthy, normal. The other one is sick. So all these pharma companies in the whole world, take all these multi-billion-dollar companies, and also hundreds of various research departments in the whole world. You can take it different names like pharmacology, physiology, oncology, immunology, pathology, whatever be the name. But they want to work with a particular protein to identify the problem. And the idea is sick, healthy. If you're healthy, no problem. If you're sick, how do you fix and get it normal? That's the way. Okay, before we go for that, what is a human body, right? So all the living creatures, organisms from bacteria to human, plants, animals, everything is nothing but made up of cells. So I assume that our body is nothing but trillions of cells. They say 40 trillion cells, 300 trillion cells, bunch of cells. And each tiny cell has 400 million proteins. So our body is nothing but trillions of proteins. So for me, I just consider trillions of proteins sitting there, trillions of proteins sitting here, everybody. As long as these proteins do the job, we are all healthy and normal. The moment when some proteins do not do the job or do something else like a villain, then we get sick. For example, recently they identified one of the proteins called 14, that's like a T cell associated protein. The job of the protein is to identify any tumors inside the body and try to immediately remove and kill them. But due to some genetic mutations, this PADI 14 protein, becomes a villain. It supplies oxygen and nutrition to the tumor, and instead of suppressing and killing the tumor, it helps to grow the tumor. So why this happens? Because that one particular protein does not do the job. I told you that our body has trillions of proteins. The protein should do the job, then we're happy. How do they do the job, right? Proteins are the very basic fundamental units of all the living organisms, bacteria to human. Trillions of proteins, right? That we know now. How do they do the job? Each protein has something called shape. That's called structure. We call structural biology. Each protein has a shape. The cell consists of, I said, 400 million proteins, right? Different proteins like glycoproteins, cholesterol, integral protein, globular protein, alpha helix protein. All these proteins make up the cell. And then the cell communicate with each other. They communicate within inter and intracellular communication and does everything. For example, you walk outside, you get a scratch, right? Then what happens? What happens is immediately the T cells and associated cells communicate saying that there is something wrong with the thing and try to do two things. Try to heal the scratch as soon as possible. That's the first thing. The second thing is they try to prevent any infection due to the heal. Otherwise, it propagates and ultimately ends up in major complications. So the cell communicate, but cells are nothing but proteins. I said that the proteins should do the job to make us healthy and live normal. When they don't do the job, people get sick. That's why tumor develops. A lot of complications come. Any sick is associated with proteins. The proteins do the job. To do the job, they need a shape. That's called structure. For example, you take a screwdriver. The screwdriver may be a Phillips screwdriver or star screwdriver. Or you take a saw that cuts the wood. That has to be in a proper shape to cut the wood. Or you take a hammer to nail, that has to be in a proper shape. Screwdriver, saw, or hammer, they should be a proper shape to do the job. Though there are trillions of proteins, the protein has a fold that is called the structure. Probably some of you may know when the protein is denatured, they are not unfolded. When it's unfolded, it's a polypeptide, there's no function for them. They have to fold and do a structure. Say, that proteins have fold like alpha helix, beta strands, and loops. That's all. For example, you think I have a protein about 200 amino acids, right? They fold something like this. So each is a helix. I usually bring I did a lot of teaching at different schools, particularly at Columbia University, New York, when I was part of Brookhaven lab. Every week we used to go for a meeting to New York City. New York City is a hub of several institutions, NYU, Columbia, Cornell, Rockefeller, Mount Sinai School of Medicine. There are 10 big schools are there. And the structural biology group is very big and strong. You always have a meeting, and I taught this several times. I take a box with all this, the protein structure and all these things to show them. Assume that this is a protein, right? Now, so assume this is the protein, and this is a small molecule. It has to bind like this. This is an active site. Okay. For you now, you see that is binding, right? It's holding that, binding that now. Now, you don't see any change here, but it doesn't hold it anymore because something happened in the active site, some mutation happened. Though the structure looks almost similar, one small change, that's what results in cancer, right? The mutations result in cancer. One small change in the binding pocket, active site, changes the function of the protein. So the meaning of that, they should be in the proper shape to do the structural work. That's the bottom line. Now, protein cell consists of all the functional basic units. The cells are nothing but consists of millions of proteins. Cell signaling is the one that is told about the scratch. When something happens, they communicate to each other, they communicate within them to do the function. For example, I want to pass this pen to the very last person sitting there. Then I just give it to him, give it to, then finally it reaches there. But my job is from here I walk and give it to him. That's all my job. So each protein within the cell does the job. So the whole function is complete. That's a cell signaling. That's very important for the whole thing to happen. The structural biology uses some method. There are different methods. Each of them is a very big technology. X-ray crystallography, NMR, cryo-EM to do the molecule structure. What they do is, you have to know the structure of the protein to design a drug molecule, to understand how that functions. If it doesn't do the function, why it doesn't do, how do you change the mutation back to the normal? For that, you need to know the structure. That's what the structural biologist do. So in simple terms, structural biology helps to understand detailed structures of protein involved in cell signaling. While cell signaling studies, I mean cell biology basically, how these proteins send messages within and between cells to run the whole show. Okay, there are different signaling pathways. Some of you, for example, there are different labs in the US or everywhere, of course. They work on one particular pathway, or they take few key proteins in the pathway. They work for 20, 25 years, 30 years. The whole life is just a couple of proteins they work on them. It's not that easy. You take one path. For example, I worked on several cancer research projects. I was part of human cancer genomics at New York. So where all these universities come, they picked up few targets, the signaling pathway. They took the protein and analyzed, finally tried to design a drug molecule to reverse that process, to make it work, to suppress the cancer, all this thing. So for example, MAP kinase, Tyagarajan knows that when we were in St. Jude, so our department, structural biology department, has close to 300 scientists, one of the biggest structural biology, 300 scientists, all PhDs. There is no master's program and no PhD program also. All of them are either postdoc or some scientists. 300 scientists working on various structural biology projects. The MAP kinase is one of the which I also part of that. There are different proteins are there. For example, RAS, BRAF, CRAF, MCAF, ERAF, EREC, all these proteins. So they pick up one of the proteins. They do a lot of biophysical, biochemical, lot of literature. I don't want to get into why you pick up the protein. That's the PI's interest. They pick up the protein, try to understand how that works, how that is related to cancer. Because the two diseases that kills a lot of people, one is cancer, the other one is heart disease. These are all kind of invisible, silent killers. Unless we do the test, sometimes suddenly we come to know that some people have 4th stage cancer. You are done. 2nd stage cancer. So it's kind of invisible. The other one is heart disease that also suddenly come and kills. For example, the MAP kinase pathway is related to cardiovascular disease and cancers. That's why a lot of groups all over the world work on kinase projects. The insulin signaling pathway is just giving some example that relates to diabetes, sugar related, and then related to cell growth. So type 2 diabetes, obesity, and other diseases are related to pathway. Two simple examples. So you pick up the pathway signal you want to work on, and you work for your whole life on that. That's what PIs do. Okay, example of MAP kinase pathway. Then RAS is one protein which I work a little bit. So that protein can be in an active state or inactive state. Depending on that active, inactive, can help to grow the cancer or help to suppress the cancer. Okay. So depending on if it binds GTP or GDP, it can be in active or inactive state. So why this happens? Because as I said, the protein is there. Sometimes some mutations happen. Why this happens? Because of some genetic alterations or toxicity. Something happens. When this happens, the function changes. The active become inactive, inactive become active. That causes the problem. So RAS mutations particularly prevalent in lung, colon, pancreatic, and bladder cancer. Because I'm part of the Cancer Research Center, I work on a lot of cancer-related projects like breast cancer, colon cancer, and breast cancer, colon cancer, prostate cancer, these cancers, because it's not my project. A lot of MDs, doctors are working on it. Associated clinical research, they bring the samples, but they want to understand why that happens. then it's kind of, I put my expertise to understand and analyze the protein and give them back the structural work for them. So targeting RAS protein has been a major focus for cancer therapy. A lot of people work on that because then, they how to prevent the cancer. Okay. Generally, how to understand why we need structural biology now, right? So here we have to understand there are three things are major. One is centimeters, micrometers, and nanometers. The size of the thing. For example, stomach, if doctors can cut open and see really what's happening. Is there a tumor? They can visualize that. We don't need anything. Generally, you can just visualize. Some of the tissues, sometimes they take the cancer tissues, they analyze with like microscope to understand that. But the other complications, for example, if it goes further down, if you want to understand at the molecular level, at the protein level, then the only choice for you is to go for structural biology, 3D structural work to understand the protein molecule, because we know that this human being is nothing but, for me, it's made up of trillions of proteins. As long as the proteins do the job, this guy is healthy. When they don't do the job, he gets sick. But I want to understand why he is sick. Then the protein that is, the main reason, culprit for making him sick, I want to understand that. To understand that, I have to go for this thing, NMR, X-ray crystallography, electron microscope. That's the one. I was running a huge facility at the lab that's called Brookhaven Lab. How I know that is there are major national labs in the US. One is in East Coast, that's in New York. One is in West Coast, that's in California. One is in Mid-South, that's called Chicago. They're like thousands of miles apart. The US is a big country, East Coast, West Coast, Mid-South. The East Coast, I was lucky enough, I don't know how I got into that. I become slowly growing to that level. I was able to manage two major facilities for the whole group. These labs, for example, this X-ray crystallography lab, I'm sure at the very end what's the facility. These labs cannot be established in the individual universities. It's impossible, lot of safety issues, and extensive money is involved. They are multi-billion-dollar labs. It's not a $1 million, but billion-dollar lab. The US government has spent a lot of money for research. They spent multi-billion-dollar labs, and they constructed those labs. So, for example, in East Coast, there are a lot of top schools are there. In New York itself, the city itself, there are about 10 schools are there. Columbia, NYU, Cornell, Mount Sinai School of Medicine, Rockefeller, so many schools are there. And then New Jersey, you go, Rutgers and Princeton, a lot of schools. Another hub is like the Boston area, where you have MIT, Harvard, Yale, everything is there. All these people used to come to use our facility. That was a great advantage for me because I had a chance to interact with the top people to do the work. It's like a win-win situation because they are not structural biologists, they are biologists, but they need to know the structure, they need my help. Because as part of that system, I was able to help them. Not only that, the pharma companies, in New Jersey, there's a lot of pharma companies are there. Johnson & Johnson, Merck, Pfizer, Eli Lilly, they are the huge monster companies, multi-billion-dollar. And very regularly, they used to come and do the experiment for drug optimization at the beamline. I'll explain a little bit about that. So anyway, the idea is, I want to know why this guy is sick. If it is a larger problem, I can just visualize, like microscope. If you want to understand them, really get into the core of the problem at the molecular level, I must know the protein structure. Why I have to know the protein structure? Because you have to understand what the protein is doing, why it becomes a tumor, why the protein is not doing the job it's supposed to do. For that, we have to know the structure. The structure is the fold. That's very important to know. That is why they come and use this technique, cryo-EM and X-ray crystallography and MR techniques to do the work. Proteins, what is protein? That's the building blocks of everything. So I said that all the living organisms from bacteria to human made up of cell, But cell is nothing but millions of proteins are inside the cell. So for me, everything is protein. So we have to understand the protein to do the thing. So that comes from, when we eat something that breaks down, amino acids, the proteins are, that they are made up of the 20 amino acids, alanine, valine, glycine, all these amino acids come and form a chain and they fold and form the structure. And then proteins have nutrition value, and then depending on amino acid composition of protein molecule structure characterizes, these are the idea about protein. So this is what important. So we have to understand that this is what my thought is I'm an expert in structural work. So, and then I will tell where is the binding pocket, what kind of thread molecule can go and bind to make it active, inactive, or do all kind of things. I can give an idea about that. So that's what it is. So these works can be done at national labs. It's not possible to do at individual universities. I mean, we can do the known proteins, but unknown large protein complexes, thing has to be done. Okay, then we do structure activity relationship. How a drug candidate interact with the target protein, right? So first we do the structural studies, then biochemistry activities, then mutational analysis, and molecular pharmacology. So each one of them is a big topic. In fact, I have like several slides for each of those slides, to show more, but that's not the basic idea. The overall idea is how the drug works, where it binds, how do you identify the binding pocket, and how do you optimize the drug for better binding to make. It's like in the olden times, if you get locked out, right, you call the locksmith. The guy comes with a whole bunch of keys. You remember? I don't know. You kids are too little, but we know that. He comes in the village, you call, he comes with a whole bunch of keys. You got locked out. You have to open the door. Then he puts all keys and try to open. Many keys go to the hole, but they don't open the lock. Only one opens the lock. Just like that, some of the proteins, some of the drug molecules can bind here, but they don't activate the molecule. So that's what our part to show how it activates. We have to figure out all this thing. And then once that is done, so these are all behind the screen, work. Finally, you get the FDA approval and the government approves it in the molecule. This is what it will do. But a lot of things happen behind. Okay. I talked about structure, which is a very powerful subject. It won more than 30 times Nobel Prize, including starting from the very first hemoglobin structure, including the very recent SARS-CoV. It's very useful to analyze the structural work because you have to know the structure of the protein to understand its function. The function is very important to design, why it doesn't do, why it does, why it keeps quiet. That's very important to know. Okay. This I already told, there are 3 different techniques. At least you can know the name, particularly for the students. It's important to know. PAOM is an emerging technique now that does this structural work. X-ray crystallography is a very well-established technique. And Madras University is one of the world top-class, institution like 30 years back. And they have written a lot of books. And even today, some of the top schools I talked to, Tegraj, they translated those things into French, they translated that into Russian and keep that as a prescribed book at the top institutions. Such a powerful institution, such university was, department was our Madras University department. Till today, whatever Professor Srinivasan, Parthasarathi, and Ramachandra, whatever they have found and the basic thing, right, still used in the latest PAOM technique now. The basic principle is from whatever they discussed, whatever they published, the papers at the time. Okay, this is the overall idea. Once you identify the protein, for example, you work with one particular protein where you're interested in why that misbehaves, or you want to find a solution for that to fix the problem. I'm always talking about from the cancer research point of view, because I did a lot of work in cancer research. So you want to cure for particular cancer, breast cancer. How do you suppress the tumor, right? You identify the protein. Once you identify the protein, you have to express, purify, get the protein ready. Once this part is done, right, protein part is done, rest of the part is more on experimental. computation work. That is why this needs people with some math programming skill or physics background, not just pure biology background. The first part of the to protein, you need molecular biologist, biochemist work. The later part is more on computational work you need to have. But the good thing is now most of the things are highly automated. That means once you have the protein ready, you can pretty much come to the very end without knowing much about all these programs and how to analyze the data. Similarly, this is about cryo-EM. How many minutes I talked? 20 minutes, because sometimes I talk a lot with one particular say I want to move. So I don't want to go too detailed in each of that because this is not the audience for that. You are not structural biologist. I want to give an overall, like, what is protein, why the protein structure is important, what are the techniques, why we have to know, because all the living things are nothing but protein. That's why we want to understand, very basic level. This is the latest technique that's called cryo-EM, where we freeze the protein and then finally we get the structural line. Starting from protein simple grid organization. I'm part of a few cryo-EM centers where I submitted proposals and I have access to those things. So if a protein is there, I can get the final model. Sometime, as I told you that a lot of different faculties and collaborated with me. For example, immunologist, oncologist, doctors. Doctors have no idea about what is structural biology. What they want to know is, that is my protein that we identify responsible for growth of the cancer. Can you get me the structure? That's all they need. They don't care how do you do, what do you do, but we spend time and finally give them the structure. We discuss about the active site. How do you go for designing those things? Okay, this is continuation of that. Don't worry. I'm going to skip some of the slides because it's not for that. Okay, this is what the whole project. I worked on a couple of important projects. One is called the Human Cancer Genomics Project. So that was funded like 10 years by the NIH funding. 10 years they funded that. A lot of money, millions and millions of dollars given. What they do is, it was given for East Coast, West Coast, and some for other universities. And those people, universities work on the protein target, but when they want to do the structural work, they come to my lab to do the work. So we start from protein purification. First, we select the target. The target is your protein, the protein of your interest. You want to know the structure of that protein. How do you optimize the drug, where the drug binds? You come up with the idea. After that, I help them to do all these things: characterization, crystallization, crystal diffraction, solving. Each one of them has a huge field to talk. Tyagaraj knows that when I moved from New York to St. Joe, so I conducted 5 years the workshop for structural biologist group. So each one of them I talk about an hour of lecture, and then every week we group the students and repeat those hands-on training. We do that. So each one of them long topic. So this is, once the target is selected, we go all the way to the structure. Okay, this is again, I'm going to really run a little faster to show what we can do, how important that is. I don't have time to talk about each of this protein, but simply show what are the things. So I'm an expert in bioinformatics, crystallography, and cryo. These are very important for drug discovery tools. How important? Because I told that in New York, I was extremely busy with both academics and non-academics. The companies also, like pharma companies, used extensively all these things. I was part of the whole project. So I will show some representative example from that, and then I'm going to stop. I want students to ask me questions about something about structural biology or drug discovery, anything you want to know. Okay. This is called fragment-based drug discovery. Some of the the academic departments do, and also pharma companies do. It's a very powerful approach for drug discovery because it's widely used both in academia and in industry. So this produces a high-quality drug-like molecule that can go in for the clinical studies. So a lot of steps are involved. First, you identify, you do the virtual screening. Virtual screening, there are libraries where you screen the compound for which molecule can go and bind the protein. Then you have, for example, I do the structural work. and then identify there's a binding pocket, because the protein is like, it has to have a fold to do the structure. When it fold, there's a binding pocket, because hundreds of amino acids go and wrap around and do a fold, and there will be a binding pocket. The binding pocket is called active site. For example, you take a long snake, like 10, 15-foot snake. The snake fold into all these, six like this. But the most important part of the snake is its mouth. When you, catch the mouth, when you block the mouth, you are safe. That's the active site. The rest of them are okay. That's the same thing for protein. The protein is a huge, winding fold into something, but I can identify the binding pocket, and then I initiate fragment scaling. That's a big technique. A lot of chemistry labs do that. The chemist synthesis all these protein drug molecules. For example, I start with some small, fragment to show that binds the binding pocket, and then it's not like one-man show. It's a combination of biochemist, chemist, structural biologist, computational biologist. Our ultimate aim is to get this molecule. Finally, like molecule, that's what. And then we do a lot of analysis, synthesis, chemists do that. Every time we go and verify whether it binds the protein. Some of them may not bind. There is no guarantee that the synthesis molecule can go and bind. But finally, we have something like a drug-like molecule. And I will talk a lot about that, but starting from here, you can go to the end. Okay. This is what target FDA approval is called multi-billion-dollar project for all the pharma companies. They identify a problem. For example, why colon cancer happens? How do we prevent them? How do you do that? They select the target and hit identification as a library. That's called virtual library and real physical library. How many of you know about zinc library? Zinc is a library. The multi-million compounds are there. If you have time, you can go and look at that. How many compounds are there? Because the pharma industry is a very powerful industry where you can, pharma and academics, particularly for we biologists, there's the two options. If you want to get a good job, you go for pharma, or you have to develop your own academic field. Okay, so the zinc has several million compounds. Some of the academic institutions have several million compounds, physical compounds there. For example, in St. Jude, there's a department called Chemical Biology and Therapeutics. They have, you can hear me, right? If you go there, still you can hear me, right? Okay, it's not that easy. Oh, online also. Just go back. So what happens is, they have, for example, in Chemical Biology and Therapeutics, they have about 1 million physical compounds they have. So they pick up the targets, they do all these binding assays, SPR, ITZ, and then confirm that finally they go for experimental activity. So the one in the blue, right, that's I'm capable of. That's what structural biology, not me, but structural biologists do. Once you do that, once you know the compound binds to your protein, then they go for preclinical development and then clinical studies, finally FDA approval. FDA approval is something like, what is that called? We call here, the government approves that for CDC approval for selling the drug to the common people. So that's what the whole thing do. Okay, I'm going to skip some of the slides. We don't have time. This is an excellent project I did for Department of Defense for US Army. So I don't know how many of you know, there was some issue like September 11th. Most of you may be kids. There was a terrorist attack in the US, and a lot of bombs went through. And then, because I work for federal government lab, they fund a lot of money. The botulinum is a bioterrorism target. Then they want us to identify potential inhibitors for that. So I played a major role. I also designed a toxin database for that toxin database and these compounds. We started from, for example, 1.4 million compounds from zinc database. At the very end, this done not just by me, but with other chemistry departments. At the very end, we got 3 compounds really bound to these toxins. That's a very major result. Unfortunately, we cannot publish these results because it's a US Army. We gave all the results to the Army, but that's a major accomplishment. And this one is for Columbia University, because I said that I worked on the human cancer genomics project. A lot of people worked. We've got amazing structures, several structures. I don't know whether I have time to talk about that. All these structures something related to cancer. So this is called Human Cancer Genomics Project. For example, the first one, human phospho PFK1, right? PFK1 is we determine the structure in APO form and bound with ADP, ATP, and then that shows the protein in active state, inactive state that tries to suppress the cancer. This is related to a childhood cancer called Tori. So this is all these projects related to some kind of cancer. But I've done all this structure for them. We had several interesting, nice papers with different groups for this cancer genomics project. This is from other group, Yale and New York University. We had, I really love to talk, but we ran out of time. I want to quickly show. I have several more slides to show. Basically, what we did is they come up with their protein target. They are always related to some kind of important cancers. I'm not worried about that, how they find. The PIs do that, but they come to me saying that, Raman, I want to get the structure. I help them to do the structural work. That's my part. So several important structures has been done. So this one is from Cornell University in Washington. So this is important. That's what I have done for. This is some small example that is with David Baker. So those structures are methodology paper. What David Baker did is, I'll tell a little bit about how many of you know he got last year Nobel Prize for computational biology, right? So I solved about 25 important structures for his group. So we published 7 papers together. That's a great contribution from myself to the great Nobel laureate. So what he did was, I told you that the human body has trillions of proteins. The protein has a structure fold. It has to be in that structure to do the job. For example, as I said, hammer should be hammer, saw should be saw to do the job. But he wanted to go the other way around. That means proteins to function, but he wants to go from function to protein. For example, you want to design a protein. You take the protein, it goes inside and clear the cancer kind of thing. Usually, they're always protein to function, right? But first you think about the function. This is the function I want, and you design a protein for that. It has to have a helix here, it has to have a strand here, it has to have a loop here, it has to have a binding pocket here. You synthesize the protein based on your theoretical idea. That is great idea. Some of the structures I have done for you. Okay, this is another structure we did for when I moved from New York to St. Jude. She was struggling to do the structural work. I helped her to do the body structural work. So these are all related to cancer research, and we published those nice papers too. Okay, this is part of various drug for St. Jude, and the cytochrome and nuclear receptor. Nuclear receptor is like a toxin removed from the body. So the nuclear receptor always look for any antibodies, not antibodies, foreign bodies inside our human, then they tries to remove. That's sometimes when you take a medicine capsule or something, the PX or nuclear receptor assumes that something wrong comes into the body. Basically, you're taking to cure something, but for PX or it's a foreign body, so it tries to immediately get rid of that. That is why the medicine doesn't work for some people. So we have to suppress that. So I worked on some very important compounds and found those compounds, and that's what PXR compounds. Okay, these are all some basic things, how protein. So I said that protein molecules, you select your target, that's the protein you want to work on. And then you have to prepare the sample, that's called the protein sample, that's called the crystal you have to do. Inside the crystal, the protein molecule is sitting there. All thousands of molecules are there. As a structural biologist, your job is to decode the information with the protein structure. Okay, so I established a course center at UTHSC. These are all the instruments I have, the sample preparation instruments. So if you have an important target, you can contact me. I mean, I can help you to do, but it's a long way. Just I'm saying, if you're local, right? If you're within the US, you can send me the protein I can do. But still, if there is a way for you to send me the protein, I can take care of all this process, crystallize, prepare the sample with my postdoc students and get those things done. These are things I established in UTHSC. This is about again about different type of samples. Don't worry about that. This is about cryo-EM structural work, how cryo-EM is working. Some of the recent projects in St. Jude, we have done those things. And how many more minutes? 5 more minutes, I'll finish. I want to talk about, this is the lab I work in, Brookhaven National Lab in New York. Just, you don't need to know a lot of technical details, but at least you can know that something like that is there in the world to do this outstanding, phenomenal research work. I said there are 3 labs. One is in New York, one is in California, one is in Chicago. So this is about, I cannot go. This is about 2 miles, 2 kilometer radius, 2 miles radius, a huge ring. And inside the ring, you see there is a basement, you have accelerators. What they do is, that atom has electron, proton, neutron. So you separate out the atom, you take this tiny atom, you separate out the photons from that, electrons from that. And then you accelerate that photons through various accelerators, electric and magnetic field. And you see the lines there, right? The blue, red, whatever. So those are all one lab. Each of them is a lab. There are about, I don't know, about 60, 70 labs are in this surrounding that. It's like a huge airport. So I was part of 2 of those, one major lab. So that lab itself, so the electrons goes through this, accelerate, they're ejected out. For example, you see the beamline has the ultra bright focus beamline come out. For example, say you are sick, you broke your leg or something. You go to the doctor, they take an X-ray, right? They show, what is broken, they try to fix. These X-rays are, I don't know, like 100,000 times brighter than those X-rays. 4.3 into 10 power 12 photons per second. Such a bright X-ray, tiny micro focus beams come. That's where you put your crystal, your sample goes there. Sample go there, X-ray go through that. You collect a lot of data, you solve the structure. Okay, that's good. Almost come to the summary. I skipped many slides because there's no point in talking too much about that. It's not the crowd. Also, it's not needed. Basically, what I wanted to show is the pharma companies, a lot of pharma companies, and there's a lot of academic departments, not just any department. Because when I worked for that long period, I collaborated with immunology, oncology, pathology, chemistry, biochemistry. Everybody does some structural work to understand how the protein works, because they are working on protein to understand that. The structure is very important to understand its function. Protein structure function is very important. Human body composed of trillions of cells that we know, and they carry out instruction, govern health and function. As long as they do the job, they're all healthy. When they don't do the job, or when they do something else, people get sick. That's the problem. So, cells composed of billions of proteins, which perform essential tasks like signaling, metabolism, and structural support. And then proteins and cellular processes, the abnormalities happen due to mutations, infection, toxins lead to the disease. Understanding these proteins at the cellular and molecular level is very important to design the drug. That's the most important thing. Unless you know the structure, how it folds, where is the binding pocket, what are the key residues, what is the charge distribution of the binding pocket, how do you optimize the small drug molecule to bind that spot to trigger that, it cannot be done. It's like, as I said, lock and key kind of thing. Lock is there, key is there. Key goes inside, but you cannot open that. Sometimes key goes, but it binds, it does not activate the molecule. When it activates, the signal goes to the next protein and the whole process completes. Drug development focuses on correcting these the problems with the protein mutations, often by targeting specific protein. That is why each group work in a particular pathway, insulin pathway or cancer pathway, or even inside cancer, there are several cancers pathway or there. Breast cancer, colon cancer, prostate cancer, lung cancer, brain tumor, so many of them there. You can take only one particular specific area you work for your whole life. So much to do, understand and work on that. Structural biology plays a very major role in optimizing the drug molecule to do the work. I think I almost completed. Okay, these are all the people I work, some of my principal collaborators, my formal boss, supervisors, and most of the funding came from NIH, DOD, DOE, US government. Okay, good, done. some questions from students. Basically, when we do the seminar, we have regular talks. So what they say, students postdocs should be given preference to ask some questions, and then they go for PS. Anybody can ask some questions. Couple of questions.
Thank you very much for that very enlightening talk. I'm behind you on the screen. I'm sorry I couldn't be there in person to meet you today. Uh, I'd like, I'm Nihal Thomas. I'm right now working with the scientists in the Stem Cell Center. And I'd like to probably invite you to perhaps on a monthly basis phone in and give a talk on each of these, unfathomable technologies which you've been talking about. Very enlightening and it's very inspiring as well.
Sure,
I'm a
yeah.
I'm a medical doctor, but perhaps I might think a little differently. I do appreciate the work that you're doing and all the hard work behind it as well.
Yeah, thank you.
Thank you so much. Are you here with us till afternoon?
Kind of leaving because I'm running out of time. Tomorrow I'm leaving back. So the last day I'm visiting today. After this talk, maybe another half an hour, 1 hour, and then I will wrap it up. And
no problems. I appreciate.
I'll be happy. I mean, someone is
keep in touch.
Interested to learn about structural biology because I've been doing for 30 years. I do a lot of teaching also. If someone is interested to know computational biology, structural biology, how to do that, I'm very happy to do some Zoom classes. That's very convenient. If you have audience, yes, I can do that. I'll be happy to.
We can organize something official for you. So I look forward to that. Thank you so much. Bye.
I love to do that. I'll be happy to be part of it. Thank you.
Thank you, sir. Thank you so much. It's an excellent input for us. I have a few questions based on curiosity because I actually come from background of drug discovery companies. So, as per your experience, which has more efficient, like small molecules or large molecules in drug discovery?
It depends on, because always the protein has an affinity for binding small molecule. So depending on protein, how big the protein is, there may be different binding pockets. So even nowadays they use the peptide for as a drug molecule. So they go and wrap around the protein and do the thing. So first we characterize the protein. That's very important. Characterizing your target protein is a very important thing. And then we do, first what we do the bioinformatics study because that's easier. Every time doing experiment is very expensive, time consuming, involves money, time, everything. So we do a thorough bioinformatics analysis of the protein molecule, analysis of binding pocket, charge distribution, amino acids, the binding pocket, and is there any pre-knowledge about the binding site? Then we go on target to binding site. So depending on the size and the characteristic of this binding pocket, we go and design the bind. For example, if my binding pocket is small, definitely very simple, I cannot put a bigger molecule there. The binding pocket is too large, I cannot also put a small molecule there. Of course, sometimes they bind through the water molecule, solvent interaction happen, but still it's not guaranteed. So the binding pocket, characterizing the binding pocket is very important before we go on to design.
So it depends upon the design for how it is designed, so small molecules or not. Based on
a design, right? Yeah, okay. If I showed you a couple of slides with a lot of different molecules, right? So what we do as a structural biologist, I can simply say this molecule binds here, but that is why the drug discovery is not just structural biology part. It adds a major component for the discovery. The chemist, biochemist understand a lot. For example, they design a protein, they design a drug molecule, and then we figure out that there is an arginine residue here. Arginine has a side chain with two nitrogen here, nitrogen. But the drug molecule design as a carbon here, right? Carbon atom. Then the chemist can go and optimize the carbon to change the carbon to oxygen. So there will be an interaction between the nitrogen and the carbon, and more interaction happen. So the chemist knows, the biochemist, the pharmacologist, for example, in our group, we have a very senior person called Dr. Miller. So he's 83 years old, he cannot believe. He's very actively designs all these compounds. So he designs all the compounds and then says, Raman, can you please get me the structure? I do the part. So chemist knows a lot how to optimize the drug molecule for that.
Thank you.
Hello, sir.
Yeah.
Can I ask one question?
Oh,
Two,
sure. Yeah.
precisely. Thank you
Yeah.
for the talk. I coming from a structural biology and a functional laboratory, so I'm very, I was very excited to see. Although were not very, your slides were not viewable on this mode, but although you speak a lot about the structural biology, I do understand that's your background. But even though after designing a specific protein molecule, and especially in, when you have a lot of drug pharma industry where we want to look at a specific drug designing and so on, so the importance of the structural biology, where the binding site is and the allosteric sites and so on and so forth, that is an integral part. But what would your comments be on a functional aspect? Because many a times when I design certain drugs or looking at a specific active site, at some point in time, they may structurally be fitting using specific, mathematical tools. But when it comes to the functional aspects, it may be tricky. That is one.
Exactly.
That is first
Exactly.
part. Second is, or of course, when you are designing a drug, as you said, in a lock and a key hypothesis, there may be times I don't know whether you might have come across where a lock can be opened by two keys. So you're fighting for the best molecule. So could you just share your views on that? Thank you.
That is why that's very important. That's a challenging, good question also. So once we finish that part, then there are two more stages before you go for a drug approval. That's the preclinical study and clinical study. The preclinical study, they do the, for example, they grow the cancer and give the drug and see. We have, you people, a lot of work on mouse model, rat model, buffoon, a lot of this model, they induce artificially the cancer and then give the drug to see that. The drug, whatever you come up, say that binds, that has to work on the preclinical model. Once that is approved preclinical model, we go for clinical model. Clinical model is we really give it to the patient. For example, we have cancer patients at my institution or also at St. Jude. Some patients are kind of given up. Unfortunately, the fourth or fifth stage, there's nothing to explore. You're going to die. But then we give an option. We are working on a drug molecule. Are you interested to, take a trial, and can we try on you? Because trying your medicine on human beings, the most difficult part, at least in the US, very difficult, because they can sue you for billions of dollars. Then if they agree with, patients, whatever, sign that, we give the untested, uncommercialized drug molecule to the patient. That's what it comes as a clinical study. If that gets approved, then it finally goes. That is very true. Just because it binds to the protein, the two molecule, there's no guarantee it will, fulfill
Give
your
a particular
target.
function. Yes.
Exactly. Because as we know, the cell has millions of protein inside. We don't know what signal it, translate to the next protein and what happens. That's why all the side effects happen. We think it happens, but something else may happen, because it's a very complex project to understand that. Unless you go, and many times, one of my friend is an excellent drug discovery person. He's Professor. He sends a couple of his compounds all the way to the drug, the clinical, preclinical, clinical. Unfortunately, it fails at the clinical stage. If it passes, you'll be a big shot. So it's possible. Yes, it's very true. Yes, that is why clinical, preclinical, and then finally FDA approval. Yes.
If
I
you
hope
can hold, thank you, sir. If you can hold some lectures on drug designing and crystallography, that's a sharp learning curve, but it'll be very nice to learn from you.
Sure, yeah, I can be happy to do that over a Zoom, and it's not a big problem, because I've been teaching for a long time. I love to teach. We do a lot of workshops also. Maybe next time if I come, I can even do a structural biology workshop. We can try some computational modeling, drug discovery, drug optimization we can do.
That'll
Because
be
as I
fantastic.
said, the protein is the basic fundamental units of living things. If you can understand the fold and the thing, that's good. Okay, good.
Thank you.
Thank you.
So nice. So I have some basic questions actually. So when you see a protein,
generally computationally. I imagine it as that surface, mountain-like surface, you'll see a lot of cavities. So during my bachelor's, we're just working on a project, and then I wanted to know the active site. I had no idea that time, and generally people will say you check the literature if they have marked the active site. If not, you do a blind docking, and there are so many algorithms. You try to see each time you run a docking algorithm, you change some parameters, you get a different active site. But for a protein, the actual active site would be one if I'm right.
Yeah.
So if you don't have the X-ray data or a crystallized data with the drug, how do you make sure that this would be an active site to design a biologic?
It's only an assumption. That is why always the docking, I have done a lot of computation work also. The docking studies have limitations in the sense, as you said, you can dock any small molecule into any site. If you define a box that you dock the molecule there, it will go on docks, give all the parameters, and then you can finish with that. But is it true? No. Then you have to go for the experimental validation for that. So if you have no idea about that binding pocket, there are cavity analysis. There are programs that analyze the various binding pockets, and then they say with this protein molecule, this is the biggest binding pocket. This is the binding pockets. These are the key residues in the binding. Gives complete detail about that. It lists 1, 2, 3, 4. You can go for any number of sites. It gives number 1, right? Then you take the number 1, and then you try to dock your molecule using all these databases. And that is not the end. That's just the beginning. Then you have to have the protein. It's very important. If you see that 2 things are needed, you should have the protein, physical protein, and also the compound you're docking with that. Then you have to mutate that key residue that shows that the arginine binds to this. This arginine has a large side chain that helps to bind this protein. Then you mutate the arginine to alanine or glycine. Then you cut off the side chain. Then again you do the binding assays. If the binding site is right, then you should see the binding. You lost the binding because you mutate the residue. First you do with the original protein, then you do with the mutated protein. Original protein, you see some binding peak. In the mutated one, you lose the thing, then that confirms the binding. Of course, that's also the initial result, not the final one. The very final one, if you want to go for drug discovery, drug optimization, you have to definitely go for structural work to identify that. That gives the real 3-dimensional view of the protein molecule with a bound wrap.
So let's say I'm working with protein. It's not a drug actually, it's protein.
Protein complex.
And there's a binder that is also peptide. And there's a structure available, and there is a paper also showing these are the 2 key residues binding. And it's on PDB. I download the structure and I see in the software it's difficult to see what are the key residues other than this that can possibly key interactions that can possibly be there. Like if you use some tool, they give you a list of interactions.
Yeah, that is very simple. There are a lot of programs are there in structural biology. I can tell you. So for 2 given molecules, protein or peptide, protein peptide, that can list you all the interaction, not just key interaction, even hydrophobic, hydrophilic, ionic interaction. All these interactions will be listed. And then if it is a large peptide, you know that it's not just, at one point, it may latch at different points of the protein molecule. Then you can go and mutate that and see what the binds are. It is possible. In short, yes, you can get the complete detailed interaction between the protein and your peptide or protein and the drug molecule. Yes, it is possible. Programs are there.
One more question. So let's say when you design a binder, any sort of binder can be a drug also. It is binding perfectly to the active site, but it is not activating the protein. So molecularly, what sort of interaction that actually shifts the so when your protein is active, maybe you'll have some sort of conformation change and it will do some function. So you have a binder that binds perfectly, but it doesn't activate the protein. So what interaction is lacking that happens?
No, that's the nature of the protein. We have to use different activator to activate it because each protein has a very special characteristic. That is why before we work on the particular target, people do extensive literature study on it because all over the different world, people take the protein work on that. The most important thing is we have to do extensive literature survey to understand the protein characterization. Then you go for it. That's very important.
So So is it like a new, let's say in the binder I added one residue that leads to formation of a new bond, maybe a hydrogen bond or maybe a chemical bond, covalent bond. So is it the bond energy that is causing the conformational change if it is active? What causes that shift from inactive to active, even if the binder is binding properly?
Yeah, again, so I showed you some example, right? In one of those cancer research projects, the protein molecule, when it bind GTP to GDP, basically same molecule, one triphosphate or diphosphate. So the diphosphate is inactive, the triphosphate is active. If you look at the structure, maybe the extra phosphate, right? It's maybe binding to few key residues. Diphosphate, usually the triphosphate is longer, right? You have one more extra phosphate. The residues that attach to the phosphate, the third phosphate is key for activating those things. So those are the key residues. You have to understand that. So then you know that you have to target on that region, because that is the one causes active to inactive state. It changes the behavior of the protein. So those things needs lot of characterization, that biophysical, biochemical characterization to understand that. Structurally, you can only show it binds or not binds. But to make sure that it shows the desired activity, you have to go for biopsy. That's why we go for preclinical study and clinical study to make sure that our hypothesis is right. It really suppresses the protein. I worked with several people. They come up with a new drug and then they are so excited, and then they inject on mouse model, and then they grow the cancer. I have seen that they grow like bigger every week. They take all the measurements, they take the picture of that, and then they give the drug. If the drug works, then the tumor starts to shrink. That's an amazing result. That means the drug really works. Your hypothesis is right. They do all these animal model studies, everything. That part is their expertise to do that. As a structural biologist, I can only say molecule binds or not binds. My job is done. Of course, you have to. That is why I said it's not a single man show. Biochemist, chemist, and molecule biologist, whole group work finally end up at the thing. But this is a very major component, because you completely characterize the protein, give the three-dimensional view of the protein molecule, talk about the key residues, the charge distribution, the binding part, everything you visualize to go and do the work.
Just to add on that, there will be a family of other protein core factors, core receptors also, that will be responsible for the activity of a specific protein or an enzyme that you're looking for.
Exactly. Yeah. That's why we have to do a very thorough what is it called, literature work. For example, when I said I showed so many structures, and I worked with 500, I deposited 500 structures with my name on that. I worked on several structures, but unfortunately, I'm not an expert in any of this biology behind that. If I start to focus on the biology, I can do only maybe 3 or 4 projects, 10 projects maybe. I cannot work on 500 structures. As an expert, you come with your idea of the project, I can tell you, okay, this is structure, this is the molecule binds, my job is done. Then you go to the other experiments to do that. A little bit knowledge in other area, but that's why we collaborate with other people.
So how much time it takes current time to resolve the structure?
It's not difficult because it's very highly automated now, particularly the one at the New York is the latest technology. Okay, that's fine. So the NIH has spent a lot of money to develop a high throughput beamline, in the sense you can literally do like thousands of compounds with your particular target, thousands of drug molecules. We don't know which one binds, which one activates. That's why we try all this thing. Can be done in about a week's time. Like 10 years back, it's a nightmare to work with hundreds of compounds. It's very highly automated. So one small example is when I started my career, so we used to do one experiment for 6 hours or 7 hours. You open the hatch, you go into the hatch, you put the sample, you align the sample, you set up all the parameters, you lock, go back and start the experiment. If something is wrong, again, you have to go back. Because the radiation, it's like more physical work. Now it's not like that. You send hundreds of compounds, they open the experimental station once, they go inside, load all the samples in the robot, and then you are done. You just start working from anywhere. You don't need to be physically, you don't need to be at the lab. So people remotely connect. For example, even you can do from here. You send a sample to wherever beamline. People sometimes, they send it to European facilities or Japan or somewhere. You send a sample, and then they give you access to the system. You sit from your home institution, you can do the experiment. So what I'm saying is highly automated. It can be very quickly done.
Let's say if I give you a protein structure and I want to know the key recipes, you're saying it will take like 2 weeks. 3 weeks.
Experimentally, right?
Yeah.
Yeah, experimentally, because the problem is not 2 weeks. It take more than that, because you have to express and purify the protein. That's a big step. Lot of optimization, you have to express the protein in E. coli, maybe in insect cell, maybe in mammalian cell, and you have to see which tag you have to add. You have to be an expert in protein production, protein synthesis. So I used to do some work, but I'm not an expert. That's really getting the sample, because it should be highly pure, should be no impurities. That takes a lot of optimization, expertise needed for that. Once the protein sample is ready, other step can be. That's why I said there are two parts in structural biology. One is the first, the molecular biology part, where you work the protein synthesis to do everywhere. The second part is structural biology part. The first part, in fact, in each structural biology group, 4 or 5 people just work on the protein production. They don't do anything. They, clone, purify, optimize all these procedures, steps to get a pure protein. They do.
Pure protein, but what I want to know is the complex.
Yeah, if you give both pure protein, it can be done in 2 weeks, maybe a week's time. It can be done. If we can use lot of parameters depending on how big is the protein, is it a membrane protein, is it how large, all these things, it can be done. At least we can make a sincere attempt in 2 weeks. Sometimes it may not form, you cannot get the structure, but still you can finish the whole experiment in about 2 weeks' time.
Last question. So recently they launched with AlphaFold 3, and they have shown that more than compared to the existing docking softwares and all those things, it's very good. So what do you think?
AlphaFold is okay, but only the core structural biologists to know that it's not something you can depend on. For example, yes, it is definitely much better than what you have. I tell you one small thing. What is AlphaFold? A protein has a structure. Structure means fold into a proper shape. Protein has amino acids like that. For example, depending on size of the protein, there may be 200 amino acids, 300 amino acids, 1,000 amino acids. These 1,000 amino acids come and fold into a proper shape. That's the protein structure. All these structures have been determined experimentally, either X-ray crystallography or cryo-EM or NMR. Different techniques are there. They determine the experimental structure. But when you don't have the experimental structure, people go for AlphaFold. AlphaFold has to use the experimentally determined structure. If there is no experimental structure, AlphaFold is 0. You can predict something, no, it won't work. But what they do, say your theoretical model has about 800 amino acids. You want to find the structure of 800 amino acids. Then it goes and do all these algorithms search against the existing experimental model. Then out of these 800 amino acids, it finds structure for 600 amino acids. Experimentally determined that matches, because there's always sequence to structure similarities there. If the sequence is same, structure should be same. That's called the homologue. Then it models based on this experimental model. But there is no structure for the 200 amino acids. Then what it will do, it will put some loop, random loop here and there. That may be an important part where you may have a binding pocket. So it is not, it's okay, but not the foolproof method. You have to be careful about AlphaFold. Okay, good.
My question is, to continue with Anand's question, there are many AI tools nowadays popping up saying that your protein structure could be this much accurate, let's say 90% confidential or 95% confidential. And there are many AI tools where we can able to mutate a particular amino acid wherever we want, and the scores are much higher. So how much we can able to rely on this?
Okay. So when you do the any theoretical model building, right? You're right. They give something called confident, is it called one parameter that shows how confident that region is. There's parameter for that. 1, 9, 0.9, 0.8, 0.6, something like that. If it's close to 1, that means it's more confident based on experimental model. Your region of interest, if it is more close to 1 or 0.9, 0.8, of course it is right. Sometime when there is no experimental model available in the database, then the confidence parameter is like 0.4, 0.5. They also show in red. That means that is not confident. You have to take a chance. It's definitely there's a wrong reach.
So we can't relate.
Exactly, we cannot rely on it. That's why you have to go for experimental model. If you're lucky, if your region of interest is more with the the confident region, yes, you can go and experiment that, you can mutate, you can do all this thing. But if it is in the parameter is low on that particular region, you cannot start. It's not there. So always the AlphaFold model depends on experimental model. If there is no experimental model, this is really questionable.
Very insightful talk. I have a very basic doubt actually. So we are trying to purify protein. It is more protein resistance. So we can see that the protein is overexpressing, but we are not able to purify it and then stably express it over a long period. I mean, leave putting resistance structures to identify the structure, but what is one way where we can identify other interacting domains of this protein? We know that it forms a polymer and it interacts with the DNA, but we're not able to identify it properly. So is there any way where we can?
There are some databases where you can do the bioinformatics, particularly if you send me the sequence later on. I'll do some work, maybe I will send email to and guide you through that. There are some. Proline is a problem that protein purification going that always depending on where it sits, it can affect the fold. Then if the protein doesn't fold, you don't get the good expression. That's the problem. So we can analyze that using some bioinformatics tools and get an idea. If you send me the sequence later on, then I will take a look at that. Okay.
Very good talk, sir.
Thank you.
Learned a lot. I think you're a very excellent teacher.
Oh, thank you.
To tell in a complex thing in a very simple way. I have one question is like how you can predict function based on structure. For example, you are a structural biologist, you can crystallize, you can do NMR, you can do all these things. But can you predict what will the function of that protein? Suppose you find a novel protein.
Okay. It is possible. You can predict. It's only prediction. Prediction can also go wrong, can go right. So prediction is possible if there is a homolog structure with more detailed information. Because one of the good thing is if the protein sequence is same, for example, it's 90% same. So already has someone worked on certain protein. For example, we worked on botulinum toxin. There are 7 serotypes, botulinum A, B, C, D, E, F. We solved only like 3. We solved botulinum A and botulinum B, for example. Then we do the sequence comparison, and then we see that 95% of the sequence is identical. The known good thing is if there is a sequence similarity, there is always a structural similarity. Then you can predict, it's only prediction again. Then it can follow like that path. The protein can also, the botulinum C and D can also behave like A and B. But there may be always some minute differences, but overall you can say predict that. It's only prediction is possible based on homolog model. If there is no model, it cannot be done. It's difficult. So function, you have to do functional studies to biophysical, biochemical studies to.
So what my question is, sir, why we are not able to predict function? We have to do a lot of biochemistry experiments, like whether it's an enzymatic activity, whether it has other interaction, whether it has signaling function, membrane function. By just doing that structure, like if we know, okay, this is a house and people are living in this, we can predict what function it has. why we are not able to predict function just based on the structure of the protein.
Exactly. That's what David Baker tried to do. Can you design a protein based on function? So we know protein does a function. Can you go the other way around? That's what very innovative, idea. But the problem is the cell has millions of proteins. We work on particular protein, but always this protein interact with other protein. It is like we know that our boy is a good boy. When you put them in the school, depending on with whom he associates, sometimes they don't behave well. We don't know what happens. We think he will return back as a good guy. There's a good chance he may also go back. So it's all the thing. What I think is depends on how it signal goes to the other surrounding proteins. How it communicate with the other proteins. Does that influence back to change this one? That could be possible.
So the other question was like you mentioned, for example, if we have, we need to purify, we need to clone, purify, and give you a pure protein so that you can find out the structure and then do the bioinformatics. Now, these proteins does not exist in isolation, like you said, in the cell, in the environment, whether they interact with other protein or DNA or membrane or lipids. So the question is like, when you have complexes, can you crystallize complexes and then see with whom they are interacting, how they're interacting, or do you need to have a pure protein or complex of protein also can be analyzed?
Not necessarily. The cryo-EM technique is very useful. You can do the structure of the complex. For example, 4 or 5 proteins, whatever the complex together, you can do the structure. You purify the protein as a complex with all these, binders, you can do the structure also. For crystallography, you need to have particular protein complex or 2 or 3. You have to characterize very well and then you do the thing. But for cryo-EM, yeah, you can do the whole the protein as a complex, like 3 or 4 proteins together. You can do it. Once you do the structure, then you can go for your interested region and then analyze more on that.
And how difficult it is to crystallize membrane proteins? Because I have heard from, during classes from crystallography teachers and all that, it's very difficult to crystallize these membrane receptors and things like that. We can do a lot of cytoplasmic proteins and all that.
Yes.
So what is your experience and why it is difficult to crystallize?
It is difficult. Membrane proteins or the solubility is very low, and I worked on membrane protein. It's very difficult to crystallize. Depends on several parameters: how big the protein is, and what are the associated proteins, how large. For example, some protein complex or protein DNA complexes are little difficult. The size matters a lot because bigger protein is difficult to crystallize. The solubility, issues are there. That is why the cryo-EM technique is there. It's very useful. We don't need to crystallize the protein. Once the sample is ready, we can freeze the samples to the grid and we can solve the structure. Yes, the answer is yes, it is difficult to crystallize membrane proteins. They don't crystallize well. And also, even if they crystallize, they don't diffract well. And of course, now the technology has improved a lot. We have the micro-focus high flux beams. Even with low resolution, you can solve the structure. But like 10-15 years back, it was very difficult to do those membrane protein structures unless they behave well. They fold well and crystallize well.
Thank you very much, sir.
Thank you.
To continue, like bicells have been used for crystallization.
Which one? The
Bicells.
yeah. They use different. What we do, there is a crystallization screen. There are centers. They have thousands of different cocktail mixtures. Once your protein is ready, we can set them all. That's why I showed that the robots and robotics are there. You put your protein and you use all these commercial screens. You set up thousands of conditions, and then you keep monitoring how that goes. And then if you see then what we do, we look at all these 1,000 drops to look for any crystals or any precipitant or something. Then we pick a few conditions, we go and further optimize those conditions. There's a long way to go, but it can be done. So we start from commercial screens.
You talked about primary cannabis site. What are allosteric interactions, drug-protein interactions?
It is also possible. We can look at that.
So
Yeah.
how we predict when we are only looking at primary
Prediction is always, as I said, there are a lot of programs out there, bioinformatics programs. You give a protein structure, they analyze the structure, they do something called cavity analysis, and then charge distribution, binding pocket, they give all this code. They give an idea where it can go and bind. But do the compound really go and bind? That we don't. That we have to do the experiment to analyze that. Either NMR, cryo-EM, or X-ray crystallography to do that, or binding assays. If you think some key residues, for example, we are working on some very intrinsically disordered protein. We screen a lot of drugs, but we know that by ATCSPR, this drug molecule goes and binds to two cysteine, residues in the protein that go and anchor it to this protein molecule. Then we know that that's the region where it binds. Then we can go and model that. So you have to have some information about that.
Based on your experience, how critical these allosteric interactions which are really there?
Activity, again, structurally, I can say that binds or not, but just because it binds doesn't mean it can involve in activity. That part we have to do the experimental part. That's really a question mark for. That's why you go for the other component.
So what's the frequency of getting a good crystal? And how do you say that this is a good crystal? Maybe in my complex, there may be some
Yeah. As I said, once the protein is ready, protein purification expression is done, protein sample is ready. And there are also commercial centers where you send a sample, they set up thousands of conditions, they send you the result. Out of thousands of conditions, they say your proteins crystallizes in 2 or 3 conditions. Then you try to repeat at the lab. You can go and optimize. What is optimization? For example, they say that you have some crystals where the cocktail solution has 2 molar ammonium sulfate, 30% PEG 3350 or something. Then you go and change a little bit that, the concentration of ammonium sulfate or PEG or pH, something you play around with that. This gradient, we do all this, keep one constant, change the other one. Then suddenly at one condition, you have nice, beautiful crystal, amazing crystal. But there is no guarantee that the beautiful crystal will diffract well. Sometimes they do really bad. In the sense, they diffract 5 angstrom, 6 angstrom. There's a limit. You have to have close to 3 angstrom or better than 4 angstrom to solve the structure. But nowadays, what we do, a lot of techniques are there. We can collect multiple data, say, combine and do that also. But it should be reasonably diffracting. Just because it forms nice crystal doesn't mean that it will give you a nice data. Also, I have seen several times, sometimes you get very ugly, crappy crystals, but that really diffract well. So unless you put them in the beamline, do the experiment, there's no, and also there are a lot of skill needed to analyze those kind of special challenging crystals, experience needed for that. So a lot of questions I like that. Back home 2-3 hours and then we all disappear. That's all I'm done.
So on behalf of CSCR, we'd like to thank you for coming over in person and then make it very simple to teach us about the structural biology. And we hope we'll continue interacting.
Sure, I'll be happy to be part, and particularly any students want some information. Competition biology is very easy because you don't need to have experimental thing. I've been doing for a long time. I can tell whatever I know. I can have a lot of access to the servers and tools I can analyze and review, but I'm also super busy, of course. But definitely I will keep your thing in list and I can give you some feedback on that. Experiment I love to do, but a lot of bottlenecks because we need pure sample and all the drug molecule, all this thing you have to ship to the thing. That's not possible. We can say, but very difficult. Thank you. But I'll be happy to help whatever things. Thank you.